We are going to go over how you're going to solve Hardy-Weinberg Equilibrium genetics problems. And I will do a guided kind of solving in the beginning after I go over how the general process works. And then we'll do a problem set together where you will check your answers after you do each problem with me. And then lastly, you will be doing a problem set on your own, which I will kind of start you off with. So if you could open up the document, that is this one, which is the, I'm pretty sure it's going to be the Hardy-Weinberg Equilibrium like study guide. And I have an annotated version that I've already written and scanned for you guys so that I'm marking down the parts that are kind of interesting or things that you need to think twice about while you're working through problems. So I just want you to kind of go through and mark down the notes that I've marked, or you can use your own terms or whatever you think is important as you go through. So the best way to gain an understanding of population genetics is to work with it. So the following information is intended to get you started on problems, and we will work through the extra set of problems at the end. So for basic information, allelic frequencies are things that we're going to be working with throughout the entire problem set. So we can express the frequency of alleles in a population numerically. Suppose we have 100 alleles of a particular gene in a population, with 70 of these being allele capital A and 30 being allele lowercase a. Well, we can calculate the frequencies, and we're going to be using frequency / ratio, whatever you want to call it, throughout almost all of Hardy-Weinberg equilibrium. So it's important that you understand how to make them and how to get back out of them. So how to make a frequency is you're going to take the number of that allele and divide it by the total number of alleles. So even if they didn't give you the total as 100, if they tell you 70 and 30, you can also add 70 and 30 to get your total. So 70 divided by 70 plus 30, which is 100, equals 0.7 or 70%. Okay. Same with the frequency of the lowercase a, you're going to be going from 30 divided by 100 to 0.3 or 30%. We'll usually stick to this ratio decimal number rather than the actual percentage, because you'll convert everything back to the decimal in the end anyways to do these calculations. So the expression P plus Q equals 1 is super important also. It's one of the key formulas that we will be using. There's only going to be 2 in Hardy-Weinberg equilibrium. So this one's the easy one to remember. So in population genetics, the symbol P represents the frequency of one allele and Q represents the frequency of the other. Thus, the preceding example, P equals 0.7 and Q equals 0.3. We can then use the expression P plus Q equals 1 to represent the relationship between the two alleles. Since all individuals in this population either have an A capital or an A lowercase allele, the two frequencies must add up to 1, right? So if there's only 2 choices in a population, if you add up all the first choice and all the second choice together, you're going to get the total number of the population. And when you're working with percents or frequencies, it's always going to total to 1, or it's going to total to 100, right? 100%. So that's the logic behind this. When we know the frequency of alleles, we can calculate the frequency of the various genotypes they will produce. So here's a diagram of how you would do a Punnett Square version to explain what is happening when you're finding the number or percents of each genotype based on the allele combinations. So it's the same way as if you have 2 heterozygotes, and they come with numbers this time because you actually know the percentage of chances that this allele is going to be passed down. And so we have a 0.7 capital A and a 0.7 capital A on both, and a 0.3 lowercase a on both. Now, when you multiply them together, you're going to be getting a 0.49 capital A, and that's shown right here because you multiply the 0.7 capital A with a 0.7 capital A, and that gives you the frequency of 0.49 for genotype AA, capital dominant heterozygous, homozygous dominant, brain fart. So if you go ahead and do the heterozygotes, that would be 0.7 capital A times 0.3 lowercase a right here. And that would give you 0.21 capital A lowercase a. But there's a second one over here. So that's going to be 0.7 capital A from here on this row. And this column 0.3 lowercase a. And then you also get 0.21 capital A lowercase a for heterozygous. So technically, when you do your heterozygotes, you would be adding them together because capital A lowercase a, whether it's 0.21 here or 0.21 here, it's the same genotype. So you would add them together in the end. And then lastly, we're going to look at our homozygous recessives, and that would be the 0.3 lowercase a times the 0.3 lowercase a in this box right here, the fourth one, and 0.3 times 0.3 is 0.09 lowercase a. So the Punnett square tells us that 49% of the population will have the genotype capital A, 21 plus 21, or 42%, will be heterozygous, or capital A little a, and 9% will be little a, which is the homozygous recessive. So that's why these equations are super, super important. This is basically a play on this equation down here, right? Because you're multiplying the p's with the p squared here, the pq, the pq, so it's 2pq, and then the q squared at the bottom for the two recessives. So that leads into the Hardy-Weinberg equilibrium equation. So the same results can be attained algebraically by the means of a formula, which is p squared plus 2pq plus q squared equals 1. So here, p squared is the frequency in the offspring of the homozygous dominant genotype. 2pq is the frequency of the heterozygous genotype, and q squared is the frequency of the homozygous recessive genotype. The important thing to note is that these are genotypes. So when they talk to you about descriptions that are physical in problems, they're going to be talking about phenotypes, and those don't directly translate to the dominants, right? So if you're talking about a heterozygous dominant or a homozygous dominant or a heterozygous, their phenotype is the same, even though their genotypes are different. So it only really helps you in a problem when they give you information about the homozygous recessive phenotype, because that automatically means q squared. And then that's an easy solve to find q. Okay, so here again, they're showing you the calculations for where it came from. So the 0.7 from the capital A frequency brings a 0.49 frequency for the homozygous dominant, the heterozygous, and the homozygous recessive, and all of it adds to 1. As shown in your text, the allelic frequencies in the offspring generation are the same as they were in the parent generation. The population is therefore said to be in genetic equilibrium. This means that allelic frequency is not constant from one generation to the next. So you don't have to have a constant frequency of your alleles or the same exact number from generation to generation in order to be in equilibrium, as long as you are still following the p squared plus 2pq equals q squared equals 1. If that still works out, then you are still in equilibrium. So this is how we are going to go ahead and solve problems. This is a little bit of a run on right there because I was writing some comments. So here's the annotation. We can use these two equations, p plus q equals 1 and p squared plus 2pq plus q squared equals 1. Those two equations are always useful in solving problems. So if it's not already written, usually in the problems, they won't write it for you, but you're going to want to write it on the pages that you're working on so that you have it at hand if you need to glance at it. So in a rabbit population, 10% of the genes for coat color are albino, which is lowercase b. So that's the recessive, while 90% are for black, capital B. What percentage of the rabbits are heterozygous if the Hardy-Weinberg assumptions are true? Well, what they gave us is that the frequency of the capital B allele is 90%, which translates over to 0.9 equals P. The frequency of the lowercase b recessive allele is 10%, which translates to 0.1, and we'll represent that with Q. So always first things first, you're going to define your P and your Q. And your P usually represents your dominant allele, and your Q represents your recessive allele. Okay, here's the equation we're going to be working with, which is the p squared plus 2pq plus q squared equals 1. So we know that the heterozygous is always going to be 2pq. So we can just plug and chug, right? We have 2 times p, which is 0.9, and q, which is 0.1. And the answer will be 0.18. Now, 18% of the rabbits are heterozygous is our concluding answer statement. If you just write 0.8, you will not get points on questions like these. Okay, so make sure you're stating what the percentage or the frequency is for what phenotype or for whatever the question is asking you for. Okay, so it's always really helpful to circle or underline in the question what exactly it's asking for at the end of the question. So again, identify what information they're providing and what they're asking for. And then sometimes you're going to decide if what they're asking for can even be found super easily or if it's kind of an indirect thing. Number 2, an example. The frequency of the recessive allele lowercase b in a population is 0.2. They gave it to you. Good info. Then a sudden catastrophe exerts a selective pressure, reducing this frequency to 0.16 in a single generation. What will be the frequencies of all the genotypes in the next generation? So the frequency of lowercase b is 0.16 equals q. Now, they gave you a recessive allele frequency in the beginning at 0.2, but honestly, they said something happened to it and it's no longer irrelevant, so you don't actually have to include that. The actual important information is the 0.16 now. We know that 0.16 is q. Therefore, using the equation P plus Q equals 1, we can do P plus 0.16 by subbing in the Q equals 1, and you can subtract it from the 1, and you can get P equals 0.84. So if you gave P, or if they gave you Q, you can always solve for the other super easily because this equation exists over here. Now, you can plug and chug it in over here now. So you're going to have the P squared, the 2pq separate, and the q squared. And after you do each of those calculations individually, you will get the genotypic frequencies in the next generation. And you can round them generally to the 100th power, or sorry, the 100th position. So your P squared equals 0.70, which is about 70%, that will have the genotype capital B, capital B. 0.27 is the frequency of the genotype of capital B, lowercase b, so about 27% of the population. And then Q squared is 0.03, so about 3% of the population will have the homozygous recessive little b, little b. Number 3, allele capital A for unattached earlobes is dominant over allele lowercase a for attached earlobes. So therefore, we're going to mark the capital A with a P and the lowercase a with a Q. If 36% of a population has attached earlobes, what are the frequencies of allele capital A and allele lowercase a in this population? So the question is asking for the allele frequencies at the end. They're not asking for genotypes, they're not asking for phenotypes, they're asking you for allele frequencies. Okay, they tell you 36% of the population has the genotype, has the phenotype of attached earlobes, but we know that this phenotype is recessive. So it has to be homozygous recessive in order to show this phenotype. Therefore, we automatically know that 36% is 0.36 equals Q squared. Okay, so in order to find Q, you just square root it, right? Super easy calculator function. Square root of 0.36 is 0.6, which gives you Q. And if you have Q, you can find P, right? So P equals 0.4. And you already found Q earlier, just in order to find P, so you have your answers. Allele T, or capital T, for the ability to taste the chemical called PTC. It's a bitter chemical. It's in a bunch of foods like broccoli. It's the same reason why some kids think broccoli is really, really gross, because it's super bitter to them. Being able to taste this chemical is dominant over allele lowercase t for the absence of this ability. In a population of 1,000 individuals, 750 are tasters, and 250 are non-tasters. What are the frequencies of allele T, capital T, and allele lowercase t in this population? So we're going to be finding our percents and our ratios first, because numbers have no use in Hardy-Weinberg. Okay, so tasters are going to be 750 divided by 1,000, because 1,000 is the total, and non-tasters are 250 divided by 1,000. Which one's more useful? Well, we know that tasters are the dominant phenotype. However, the dominant phenotype is going to be P squared plus 2pq, so it's kind of a hassle to work with. On the other hand, the non-taster phenotype automatically is Q squared. So we can actually work directly with Q squared and 0.25. Okay, so over here, 0.25 is Q squared. If you just square root it, you get 0.5 is Q. And now P is super easy to find because 1 minus Q, or 1 minus 0.5 equals P, and that is 0.5. Okay, so there are more problems. And what I'm going to ask that you do is that you are going to work on these on your own. And then I will show you the actual answers so you can check them so you know that you're doing them right. At the top of your sheet, or of the top of the handout that you are currently working on, it doesn't have to be on this paper, it can be on your own notebook. Make sure you always write down the two equations, P plus Q equals 1 and P squared plus 2pq equals Q squared, plus Q squared equals 1. Sorry, that's totally my bad, but it's on the screen right here. P squared plus 2pq plus Q squared equals 1. Always write those two at the top of any Hardy-Weinberg problem, because you're probably going to be using them and you don't want to have to try to recall it in the middle of a problem when you could already have it written down and referenced while you have a clear mind. Okay, so I'm going to ask that you pause and go ahead and work on all of these problems for the rest of this handout, and then you're going to come back to this video and check your answers. Honestly, if you decide not to do these on your own, that's not going to help you at all, and it doesn't hurt you to try them on your own at least before you check them with these answers. So I highly suggest that you are going to be doing a problem set afterwards that I'm not going to help you with. So you're going to need to get used to doing them. And this is a good chance for you to take that. So pause and go do your questions right now. All right. So here are the answers that we're going to go over for these. So for number 1, it says the frequency of 2 alleles in a gene pool is 0.1 a, capital A, and 0.9 lowercase a. What is the percentage in the population of heterozygous individuals of homozygous recessive? So the question is directly asking you for the heterozygous answer and the homozygous recessive answer. And they gave you the initial information of capital A equals 0.1. So we can automatically write that as P, and lowercase a equals 0.9. So we can automatically write that as Q. We usually rely on the letter capitalization to determine if it's dominant or recessive rather than the weight of the number. But if they give you information, they give you information. You would just go with it. If they give you the alleles, though, it's super easy to find the heterozygous because the heterozygous part of the equation for Hardy-Weinberg is 2pq. So you plug in chug, so 2 times 0.1p and times 0.9q, and then you get 0.6 are going to be heterozygous, so 36%. And then you can also easily calculate q squared, which is the homozygous recessive. And that's going to be 0.9 times 0.9, which is 0.81, or 81% are going to be homozygous recessive. Number 2, the allele for white wool is capital B, and that's dominant over the allele little b for black wool. In a sample of 900 sheep, 891 are white and 9 are black. Estimate the allelic frequencies in this sample. So we're going to be looking for P and Q. So we know that we're going to represent capital B as P and lowercase b as Q. And we also know that the coloration is right now going to be 891 divided by 900. And that's going to give us 0.99 dominant phenotypes, which is represented by P squared plus 2pq. So it's not too helpful. But on the other hand, we have 9 divided by 900, which is 0.01. And that is actually directly represented by q squared. So if we want to use that, we're going to be able to just square root that to get q, which is 0.1. And then you can find p, which is 1 minus 0.1 and 0.9. So in a population that is in Hardy-Weinberg equilibrium, the frequency of the recessive homozygote genotype of a certain trait is 0.09. What is the percentage of individuals homozygous for the dominant allele? So they're going to ask us for p squared at the very end. So we know that the q squared, because it's the recessive homozygote, the q squared is 0.09. So if you square root that, you get 0.3. P plus q equals 1. So 1 minus 0.3 is 0.7. So p equals 0.7. If you square 0.7, then you get p squared, and that's 0.49. So 49% are going to be homozygous dominant. Number 4, in a population that is in Hardy-Weinberg equilibrium, 36% of the individuals are recessive homozygotes for a certain trait. For the same trait, what is the percentage in this population of homozygous dominant individuals or of heterozygous individuals? So this is similar to the last question where they gave you q squared because they gave you the percentage of recessive homozygotes. So 36% are recessive homozygotes, which means 0.36 equals q squared. If you square q squared and you square 0.36, q equals 0.6. P plus q gives you 1 minus 0.6 to get p, and that means that p is 0.4. P squared then is just 0.4 squared, and that's 0.16, or 16% are homozygous dominant. And then heterozygous are also easy. It's just 2pq, so it's 2 times 0.4 times 0.6, and that gives you 0.48, or 48% of the population are going to be heterozygotes in Hardy-Weinberg equilibrium. Number 5 is allele T, capital T. For the particular chemical taste, it's dominant over the allele little t for the inability to taste. At Cornell University, out of 400 surveyed students, 64 were found to be non-tasters. We know that non-tasters are recessive phenotype, which means that automatically they have the recessive genotype, which is q squared. Okay, so if you take 64 divided by 400, you get 0.16. And the reason we can't just use 64 is because it's a number and it's not a frequency. So we have to change it to a frequency. Q is going to be the square root of 0.16, and therefore it is 0.4. That means P is easily 0.6 from the equation P plus Q equals 1. Now we want to find the heterozygous percentage. So that's going to be 2pq, which is 2 times 0.4 times 0.6, which is 0.48. And 0.48 means 48% of the population at that student body are going to be heterozygous for the ability to taste the particular chemical. See, on the new page, I still wrote down the equations just because it's easier to reference than if I have it at the top. So in humans, Rh positive individuals have the Rh antigen on their red blood cells. It's just a marker on your blood cell that tells you whether or not you can accept certain blood types, while Rh negative individuals do not. Assume that the Rh positive phenotype is produced by a dominant gene Rh, and the Rh negative phenotype is produced by its recessive allele, lowercase rh. in a population that is in Hardy-Weinberg equilibrium, if 84% of the individuals are Rh positive, what are the frequencies of the Rh allele and the the dominant Rh allele and the recessive Rh allele at this locus? So they gave us 84%, and we're going to translate that into 0.84, and that represents our p squared plus 2pq. Now, we know that p squared plus 2pq is only part of the equation, so the rest of the equation has to add up to 1. So if you do 1 minus 0.84, that's 0.16, and that means that q squared, which was the rest of the equation, is 0.4, and that would be the RH recessive phenotype or recessive genotype. If you square root that, then you'll get Q, which is 0.4, and that's the recessive Rh allele frequency. And then P plus Q equals 1 is the formula that you're going to use to find what P is, and that's going to be 0.6, and that would be the dominant Rh allele frequency. In corn, yellow kernel color is governed by a dominant allele and white is a recessive allele. A random sample of 1,000 kernels reveals that 910 are yellow and 90 are white. What are the frequencies of the yellow and white alleles in this population? What is the percentage of heterozygotes in this population? So we're looking for P, Q, and what 2pq equals. So yellow is p squared plus 2pq for the phenotype, and so it's 910 divided by 1,000, 0.91, but it's not really useful. So we're going to do the white phenotype corn, and we're going to go q squared equals 90 plus divided by 1,000, which is 0.09. So if you want to find q, you got to square root that, and that's 0.3. That means p is 0.7. And using 2pq to find the heterozygotes, we find that 42% of the population is heterozygous for this gene. Okie dokie. So a rare disease due to a recessive allele, which is lethal when homozygous occurs with a frequency of 1 in a million. How many individuals in a town of 14,000 can be expected to carry this allele? So the question is a little bit of a trick question that I actually annotated out for you. Hopefully you caught it too when you were doing it, but the question is asking you for carriers. So carriers have to be heterozygotes. So they're just using different language to ask for you to find the frequency of the heterozygotes. So the frequency of this presenting means that it has to be a recessive homozygous individual that presents this disease. I mean, is a chance of 1 in a million. So 1 divided by a million is 0.5 zeros and 1. That's q squared. So in order to find q, you have to square root that, and that gives you 0.001. Okay, p is 1 minus 0.001, so it's 0.999. Now we want to find the heterozygotes, which means that we're going to be doing 2pq, so 2 times 0.001 times 0.999, and that's going to be the frequency right here of 2pq. However, this isn't super important to what the actual question is asking, because we want to look at, in a town that has a specific number of people, how many of these people could actually be carriers. So you actually have to multiply the frequency or the percent with the number of individuals in the population that you're observing. And so you would multiply that by 14,000 and you get 27.972. But when it's the case of individuals, you're going to round up or down based on the decimal, because you can't have parts of people or parts of animals. You have whole individuals. So in this case, we would round 27.972 up to 28 individuals. So that means in this town, there are 28 individuals who carry the rare disease allele, but don't actually show it. So that means that if they too happen to have kids, just by doing Punnett squares, you know that each kid born has a 25% chance of having this rare disease, which is lethal and kills the child. So that's pretty crazy to think about. So this is the practice. You're going to go ahead and start on your actual problem set, which is due next class. If you look at the problem set at the top, the first set right here is just definitions. It's asking you, what does P mean? What does Q mean? What does P squared mean? What does Q squared mean? What does 2pq mean? So I'll give you some samples. So P would be the frequency of the dominant allele, right? For example, P squared is the frequency of the homozygous dominant genotype, right? So you're going to do that and you're going to write out those for me. And then number 1 is pretty cool too, because you're just counting, so you get a visual example, and then you continue to work on some multiple choice and not multiple choice. These are part questions. And then you'll answer all the rest of these questions in your problem set, and that is due the next class that we meet.